All right, we're live. How's everybody? Good, another day. Okay, so let's go over some announcements. So I'm out of town next week. I'll be traveling. So you'll have assignments posted. So if you go to D2L, I have a folder here. And let's see, so course home. And so you'll have some assignments right there. Have to still look at those, but those should be posted by Saturday. So you have plenty of time. Those will both be due on the 15th. So we'll just resume as normal on the 15th. Check out the course outline. Still have one more exam before final. I did post assignment for today here. So let's talk about that. So what'd you think? Nice, yeah. So what you consume, like building blocks for your muscles, makes a big difference. So the type of calories and the quantity of food. In this study, they were at a pretty severe energy deficit for 28 days. So much below what they needed, at least in theory, to maintain their body mass, right? So there were two different groups compared, right? A higher protein group and then a control group. So if we look to see what the caloric restriction was, we can go down here. And so we hypothesized that during an energy deficit of 40%. So they're almost cutting their calories in half of what they would need for maintenance, which is, wow, that's not eating a lot of food. So estimated energy requirements. So maintenance would have been, as they say here, 33 plus or minus 1 calorie per kilogram of lean body mass. So that's not just your total body mass in kilograms, that's just your lean body mass in kilograms. So during lab, we've calculated that, and we will calculate that today, what your lean body mass in kilograms is, because that correlates with the fat-free mass index. Remember, your lean body mass divided by your height in meters squared gives you your fat-free mass index. So they were basing their caloric deficit not on their total mass, but on their lean body mass. Fat doesn't expend too much energy, so really it's the lean component that is burning all of those calories. So makes sense. But the higher protein group, 2.4 grams per kilogram per day. So you could, if you wanted to, calculate that for yourself. So your body weight in pounds divided by 2.2 multiplied by 2.4. Hey, Britton, come on in. And then the lower protein group is 1.2 grams per kilogram. The lower protein group actually 1.2 grams per kilogram is pretty high, considering that the recommended dietary allowance is 0.8 grams per kilogram for a non-active individual. So anytime you improve your activity level, you're going to need higher energy requirements. So that's going to come from protein. So the only difference between the groups was the protein intake. So the total calorie consumption, the macronutrient percentages were the same. If you go down further, so looking at the control group, so 15% of their total calories came from protein, 50% of their total calories came from carbohydrates, and then 35% came from that. The higher protein group looks like 35% from protein, 50% from carbohydrates, and 15% from fat. So they're swapping the fat for the protein, keeping the total energy consumption the same, which would have been challenging because how many calories does 1 gram of protein have from your nutrition class? Do you remember? Yeah, 4. But then fat has, you remember how many per gram? Yeah, 9, so it's a little more energy dense. What I really liked about this study though, and it had to have tremendous amounts of funding because they gave them all their meals. So you can imagine running this type of study, you'd have to have a pretty good staff on hand. I can just see it's probably graduate students that are being paid to distribute meals. But so participants were provided with all meals and beverages to consume throughout the intervention period, with the exception of water and non-caloric drinks were ad libitum. So can you imagine being a participant in this study? So, okay, you wake up, breakfast time, so got to get your coat on, your jammies, and you're going down to the lab to have your breakfast, and then a couple hours later, same thing again for 28 days. 3 meals a day at the research facility. So what a great study though, because most of these types of studies, it's self-reported diet, which is not as valid. I suppose they could have been like binging on cookies or something towards the end. I can see them like really tempted, but based on the results, it didn't seem like that was the case. They seemed to stick with it all the way through. So, oh, this one came up here, Paris Benedict, remember that? We did that in class. So prior to undergoing testing, they put everybody on a 3-day maintenance diet. So that's up here. So participants were provided with a 3-day diet for weight maintenance, energy requirements based on the Harris-Benedict equation with use of an activity factor. Okay, so remember that. Based on the subject's self-reported habitual daily activities. And then on day 3, after an overnight fast, they came in for all of their blood testing and body composition. So let's get to the results, which were pretty neat. They did a bunch of tests, a lot of them that we've experienced in our lab. So they did high intensity and sprint interval, high intensity interval training, sprint interval training. Session. Wow. So read this. So sessions consisted of 1 session of sprint interval training progressing from 4 to 8 30-second Wingate bouts. So that's 1 workout. So can you imagine doing that for a workout? So you're on the bike and you're saying it's not just 1 and done, it's 4 to 8 of those. So don't worry, I won't have you do that in lab. 4 minutes between bouts. So lots of high intensity cardio. Looks like full body resistance exercise circuit 2 times a week, no rest between exercises. So kind of a high volume type, 10 reps a set. Last set of each exercise to failure, 1 minute rest between exercises. So lots of activity combined with very little calories. So researchers didn't want to take any chances of not losing some body fat there. So a lot of times what happens with research is if you put people on a program where they're really doing a lot of activity for the program, then during the non-exercise hours of the day, they kind of become less active. It's kind of a compensation mechanism. There's some physiological reasons for that because our brain is fighting to maintain our body mass after a certain point, because the more weight we lose, the more difficult it is to keep losing. So to combat that effect, what they had them do is accumulate at least 10,000 steps a day. So in addition to what they were doing for training, they slapped a pedometer on their belt and they're having to accumulate 10,000 steps. How many of you have tried to do that? 10,000 steps a day? It's kind of a benchmark. It's been around for about 20 years, and research in Japan showed some significant health benefits if you accumulated that threshold of steps a day. There was one study, I'm getting off track here, but one study done on the Amish, because I used to be in Illinois where there's a lot of Amish people, so studies done on steps a day for Amish people. Guess how many? It's like 25,000 a day, and it's amazing. So just the differences in lifestyle, but they wanted to prevent them from being less active during the time outside the exercise bouts. So the big take home is depicted right here. So we're doing body composition today, and those who've already been to lab have looked at this with the in-body. We're going to be going over that sheet and all the different variables. So let's scroll up just a little bit. So the higher protein group, 2.4 grams per kilogram, versus the control group, 1.2 grams per kilogram. These are the changes over that 28-day period. So in total body mass, both groups lost, it looks like, around 4 kilograms was the change. So 4 kilograms times 2.2 would give you the amount of pounds. What I thought was really interesting that showed significance, so you can see the asterisk here to note significance, was the gain in lean body mass by the higher protein group. So we're talking about a 40% energy deficit, eating 40% less than what you need just to maintain, but in spite of that, they have this anabolic response. So gaining lean body mass is counterintuitive to what your body wants to do in a caloric deficit, right? So what we're saying is it's very difficult to build muscle when you're not eating enough. True? Yeah. So to build, you have to have the surplus available. You have to have those construction materials in your body available to build new tissue. So what's exciting is even in the presence of a 40% energy deficit, when we're consuming 2.4 grams per kilogram, they were able to build, it looks like, maybe a little over 1.5 kilograms of lean body mass. So you think about that, that's maybe like 4 pounds, a little over, maybe not quite 4 pounds. So that's significant. Whereas the control group with 1.2 grams per kilogram, were they losing any lean body mass? I guess to look at the glass half full for the control group, they maintained, which is good. That's what we want. If we're in a caloric deficit, what type of weight do we want to lose? It's not the muscle, right? It's the body fat that we want. So that's the calories that we want to be expending. So from a fat mass standpoint, looking at the kilograms lost, the higher protein group lost significantly more than the control group. So the control group still had a really good response with 1.2 grams per kilogram. We're still maintaining muscle and we're losing fat mass, which is good, but elevating the protein intake and lowering the fat intake, so swapping those two macronutrients, we're building muscle and we're losing a little bit more body fat. So I thought that was a really good message because as a practitioner, you'll be working with people that have pretty much that goal in mind. So, questions? Okay, so could I collect that from you? Because I'll forget. I'm going to ask. So that's maybe a study that you want to keep because This is one of those studies that you'll come back to show. I think I got yours. Okay. Yeah, so if you want to just send it later, that'd be good. That was a good read. Okay. So I've got a nice video on strategies that athletes use to increase their oxygen transportability. So we'll get to that, but I want to go back and talk about a few of our slides. Just the last two slides of chapter 9. So we have different factors that stimulate respiration, that stimulate breathing, okay, that stimulate this cyclical action of inspiration and expiration. So what part of the brainstem is involved with respiration? Remember? Yeah, pons and medulla. So the medulla has that ventral respiratory group that pretty much coordinates everything that activates the inspiratory and expiratory muscles. There are chemical indicators in the blood that send signals to the brain that control respiration, so the rate, depth, rhythm of breathing. And so oxygen pressure is one. So what we see here is that oxygen pressure, certainly, as it begins to drop. So as oxygen pressure is dropping, we get to a threshold of about 60 millimeters of mercury. So that's 60 millimeters of mercury, and then all of a sudden we pass that threshold, and what happens to your ventilation? Shoots right up. So it's kind of a curvilinear response where our brain really doesn't do much until the oxygen pressure in the arterial blood, so PaO2, oxygen pressure in the arterial blood, drops below 60 millimeters of mercury, and then we have this huge ventilation response. So a combination of increasing our depth of breathing, okay, so that's the tidal volume, and also the frequency of breathing. So that's one factor. The next factor, probably the most important, has to do with carbon dioxide. So the most potent stimulator of respiration is the arterial level of carbon dioxide. So good test question. So I'll ask you what three factors is partial pressure of oxygen, partial pressure of carbon dioxide, and what's the third one, do you think? Has to do with acidity. So your pH, right? pH. So as the pH drops, our ventilation goes up. So carbon dioxide in the arterial blood has much more of an influence on respiration than does oxygen or pH. For example, in the tissues, as the arterial blood goes from 40 millimeters of mercury to 45, that results in a doubling of pulmonary ventilation. So much more influential than the partial pressure of oxygen. So the partial pressure of oxygen had to decline by a lot before anything was evident with ventilation, but with carbon dioxide, if you go from 40 up to 45, look at the difference. It's a doubling of your pulmonary ventilation. So just showing support for carbon dioxide being the primary stimulator of ventilation. So with David Blaine, held his breath for 17 minutes. One of the things he talked about was practicing holding his breath. And so he would get up every morning and he would do this practice for an hour. And so he talked about hyperventilating and then holding it for as long as he could, and then resting for a period of time, then hyperventilating again. So what does hyperventilation do? He mentioned it.
It decreases your CO2.
Decreases your CO2, which would do what to your urge to breathe? Would you have a reduced urge to breathe or an increased urge to breathe if with less CO2 in your system? You have a decreased urge to breathe because CO2 is the primary stimulator of ventilation. If this hyperventilation response, you're blowing off CO2, So lesser amounts of CO2 means less urge to breathe. And so that's an advantage. So you'll notice that sprint swimmers that want to hold their breath for long periods of time because it improves or reduces their drag resistance will hyperventilate just before getting onto the blocks because that reduces their urge to take a breath. So that's all. From a safety standpoint, though, can that be a problem? It doesn't increase your oxygen. Yeah, so your body still needs oxygen. If you don't have enough oxygen, yeah, you're going to pass out. So there is a safety risk with that. Okay, so getting into chapter 10 now. So response, training, and adaptation, special considerations. So the respiratory system is a system that tends to come overbuilt, so to speak. So for normal recreational exercisers, it can handle just about anything we can dish out. Once we get to the elite level endurance performers, that's when we can have a limitation. So it's different than the cardiovascular system, the heart, the blood vessels, where we do see a lot of changes. With the respiratory system, we don't see as many changes. So this is just on D2L, just like with all the other PowerPoints. So breathing is matched to the workload. Okay, so as the workload goes up, ventilation goes up. So remember, ventilation is a combination of your tidal volume. Okay, so that's the depth multiplied by the frequency. So it's kind of like cardiac output with heart rate and stroke volume, only now you're talking about airflow. So the depth of breathing multiplied by your frequency gives your total airflow per minute. So there's a very large reserve with the pulmonary system. So it's generally not a limiting factor when you're talking about most people, moderately fit individuals. Even if you do feel out of breath during exercise, it's still not being pushed to its limits. That can change though when you have an elite endurance athlete. So an elite endurance athlete who has maximized the adaptation of their cardiovascular and metabolic systems, those adaptations can ultimately exceed the respiratory system. So they can pass the respiratory system, the reserve of the respiratory system, and then that's when you have a mismatch where in elite, highly trained athletes, you might see a little bit more change in the respiratory system to keep pace with those other systems. So oftentimes the respiratory system is paired with the cardiovascular system. So it's called the cardiorespiratory system because obviously working together to bring oxygen in that binds with blood cells that we transport to muscle. For this chapter, we'll discuss each of these three things in the context of different exercise intensities. So pulmonary ventilation, let's define it. It's liters per minute of airflow. Okay, so tidal volume multiplied by breathing frequency. External respiration, that takes place in the lungs. So right ventricle pumps blood to the lungs where we have exchange of gases. Okay. Internal respiration takes place at the tissues. So remember, the lungs have their own blood supply, as well as the heart, as well as all the other tissues of the body. That's internal respiration. So is everybody okay understanding where each of these processes take place? Okay. All right, so let's keep going. So the three different types of exercise that we will talk about, each of these three things: short and long submaximal activity, and then lastly will be incremental exercise to maximum. So we'll be doing the Bruce protocol in a few weeks, and that will be incremental, especially because It's the incline that creates the problem, not the speed. Okay, so let's take it a step further. So pulmonary ventilation, we have all these variables that relate to that. So minute ventilation is that's the liters per minute of airflow. So dead space, remember how we define that? That's the air that doesn't participate in gas exchange, right? So tidal volume is your depth of breathing, frequency, how many breaths per minute. This ratio is interesting, the ratio of dead space to tidal volume. So we'll pick up on that later on because it's easier to see if you can see it in a graph. External respiration. So this is the alveoli. So the difference in gas pressures that allows for exchange in blood that comes from the right side of the heart. So I want you to notice that when we have a capital A versus a lowercase a, so capital A is the alveoli. So partial pressure of oxygen at the alveoli. So that's going to be pretty high, right? Partial pressure of oxygen in the arterial blood would be lowercase a. And then let's see, a couple more. So SaO2%, percent saturation of the arterial blood with oxygen. As we go over here, internal respiration. So that's of the tissues, right? So the AVO2 difference. So you have the arteries, you have the veins, and then you have the tissues in the middle. So the tissues is what takes the oxygen. So the oxygen in the arterial blood is higher than the venous blood because some of that was taken up by the tissues. So that represents the arterial venous oxygen difference. So you have different variables here: arterial pressure of oxygen, arterial pressure of carbon dioxide, venous pressure of carbon dioxide, and so on. So any questions on these variables? Pretty self-explanatory once you kind of visualize what the blood is doing at different points. So we'll talk about each of those three exercises in the context of these things. Okay, so short-term light to moderate submaximal aerobic exercise. So basically, just your daily activities. Going maybe to a brisk walk. Pulmonary ventilation. So we need two things to calculate pulmonary ventilation, right? We need tidal volume, okay, so that's your depth of breathing, and we need breathing frequency. So do you remember when we talked about metabolism, how the first couple minutes, it's kind of your aerobic system is upregulating, kind of warming up. So during the first few minutes, even when it's light exercise, we're using the anaerobic systems during the first couple minutes. So what you see with ventilation is kind of like what you see with heart rate. So for the first maybe up to just under 3 minutes, you see this gradual rise, and then your body's nice and warmed up, your oxidative system's running great, and then what happens? You flatten out. So that represents the steady state. So you have this warm-up period, warming up, your respiratory system, cardiovascular system, muscles are upregulating, and then everything just flattens out. You're able to keep pace with the demand of the activity. Okay, so ventilation, just like heart rate, gradual rise and then plateaus. So tidal volume is your depth of breathing. So you can see here the liters. How many liters of air are you breathing per breath? And so just like the ventilation, tidal volume rises and then flattens out. So your depth of breathing doesn't change after a few minutes. You're able to keep up the pace. Okay, so the dead space over tidal volume. Okay, so what you see here is that drop. So What that means is the dead space is reducing while the tidal volume is increasing. So if you understand ratios, okay, so this is the ratio, right? This is the number, the ratio. If dead space is going down, that's in the numerator, and tidal volume is going up in the denominator, what's happening to the ratio? The number that you get, it's going to drop, right? As the top variable goes down, the bottom variable goes up, it's going to drop, just like you see that line there. So that just means we're using more of the oxygen. More of the oxygen is participating in gas exchange. Frequency of breathing, same response, right? Breathe more frequently and then plateaus out. So we're just describing a steady state. Okay, external respiration. Where does external respiration take place? In the lungs, yes, in the lungs. Okay, so alveolar ventilation. What's the difference between alveolar ventilation and pulmonary ventilation? What do we have to take into account with alveolar ventilation? Yes, okay, yeah. Alveolar ventilation is tidal volume minus dead space. Okay, whereas ventilation is just the whole thing together. So alveolar ventilation is tidal volume minus the dead space times the frequency of breathing. Whereas ventilation is just the whole tidal volume with taking into account the dead space 2 times the frequency. So it's basically just the same thing, minus the dead space. So it's coming up and then it's plateauing. So just indicating a steady state. So got a question for you. Okay, remember what capital A stands for? Alveoli. Okay, partial pressure of oxygen in the alveoli. What does the lowercase a stand for? Arterial, yeah. So partial pressure of oxygen in the arterial blood. Okay, so why the difference? Partial pressure of oxygen in the alveoli is 104 millimeters of mercury, but by the time the blood gets back to the left side of the heart, the pressure has dropped down to about 95. So why the difference? Remember? Yes, it dilutes. Yes, from where? No? Yes, lung circulation. So right there is what we've got. So the lung circulation, the bronchial veins are returning blood to the heart, and that blood mixes with the blood from the external respiration. So that's going to drop the pressure. So the pressure right here would be 104, but then it mixes with the bronchial veins blood, which is relatively deoxygenated. And so then by the time we get back to the heart, it's 95. So very good. It dilutes it just a little bit, is the idea.
And that's the blood that's supplying the actual lung tissue.
Actual lung tissue. It's like the heart. So the heart has blood flowing through its ventricles, but that doesn't do it any good. It has to have its own blood supply. So it's a coronary circulation. So it's the bronchial circulation that is the reason. Okay, so internal respiration, 4 reasons. This is a great test question, so you want to highlight this slide in your notes. Okay, so internal respiration, where does internal respiration take place? In the tissues, so including the lungs themselves. Okay, bronchial circulation. So 4 reasons. The 3 there are the most important. So it's interesting that the partial pressure of oxygen, yes, that is a factor. However, these other 3 are a lot more important, especially carbon dioxide. Okay, so we say it causes a shift to the right in the oxygen dissociation curve. So you're thinking, what's that? Well, let's talk about it. So this is the oxyhemoglobin dissociation curve. So let me explain. So the y-axis here, that's the percent of hemoglobin bound to oxygen. Percentage of hemoglobin bound to oxygen. The x-axis, this is the partial pressure of oxygen. So what we're saying is when the curve shifts to the right, so in the tissues, as we have higher temperature, lower pH. Lower pH means what for acidity? Means higher acidity, okay? So temperature, pH, carbon dioxide in the tissues. What happens is that we can see if we compare oxygen pressure, so for a given oxygen pressure, for a given oxygen pressure, so follow this line up. So let's say 40 millimeters of mercury, a given oxygen pressure, if the curve is shifting to the right, there is less oxygen bound to hemoglobin. So going from this black line, whether it be 75%, down to this blue line where there'd be about 65% for the same pressure of, I'm not sure why it does that, same pressure of oxygen. So what we're saying is that as temperature goes up, as pH goes down, as CO2 goes up, we have less oxygen bound to hemoglobin for a given pressure of oxygen. Okay, so does that make sense? Okay, all right, so, and that just is pronounced as we go from rest to submaximal to maximal exercise. Look at how much, look at the percentage of oxygen bound to hemoglobin. With maximal exercise, we have almost all the oxygen is unloaded from hemoglobin at that point. So it's almost down to less than 90% saturated. It's only about 10% saturated under conditions of maximal exercise. So that's a good thing because once the oxygen unloads from hemoglobin, where does it go? Into muscles, right, where we need it. Okay, myoglobin picks it up in muscles, transports it where? Mitochondria, that's where we use it to make ATP. Okay, internal respiration. So AVO2 difference. So the difference between oxygen content in the blood on the arterial and venous side. So you would expect that difference to grow, right? From rest to exercise, because we're extracting more of the oxygen. So the difference is going to be greater. You're going to have much less on the venous side. So with submaximal activity, the difference is going to change and then it's going to level off. So as soon as we've met the oxygen requirements, it levels right out. Percent oxygen in the venous blood. Why does it make sense that this is dropping? So we go through the tissues, the blood is now in the venous circuit on the way back to the heart. Why does it make sense that percentage of oxygen in the venous blood drops with exercise? Because what are we doing with the oxygen? Yeah, it's being taken up by the tissues, so of course you're going to have less in the venous blood as it goes back to the heart. So that's what is being depicted here, is the percent is dropping because we're using more of it at the tissues. Okay, so this just depicts the direction of each of these things. So CO2 we produce in the tissues. Okay, what phases of metabolism for carbohydrate? So pyruvate to acetyl-CoA, we make CO2, and then Krebs cycle, we make CO2. So we make CO2 in the tissues, and so that produces a higher pressure, and so then CO2 is transported into the capillaries, back to the heart, exchange at the lungs. So we also release hydrogen ions. Okay, so whatever theory you espouse to, if it's the classical theory, we make lactic acid, but what happens to it? Almost immediately as soon as we make it. What happens? Yeah, it dissociates, splits apart into lactate, which is harmless, and a hydrogen ion. Okay, so we have to do something with the hydrogen ions. We have to buffer those, and so those are shuttled out to the blood where we have, what's the primary buffer in the blood? Also transports carbon dioxide, 70%. Bicarbonate. Yeah. So that whole reaction where hydrogen ion binds to bicarbonate, we get carbonic acid, which then dissociates to water and CO2 that we exhale at the lungs. Heat. So, we only trap about 40% of the available free energy into ATP. So the other 60% is wasted. So our metabolism is relatively inefficient. The other 60% of that free energy is released as heat. So that's useful during some times of the year. Other times of the year when it's warmer, we have to dissipate that heat to maintain body temperature. Okay, so everybody good with the first category of exercise? Light exercise. Okay, so now we have two more categories to go. Okay, so moderate to heavy submaximal. Okay, so let me ask you a question. Are we still working in a steady state eventually? Yes, it's just a higher steady state. We haven't crossed, what's that threshold where eventually we're not steady state and we've crossed anaerobic threshold. So we're still working within the aerobic zone, but it's at a higher level. So pretty much all the responses that we saw with light exercise are the same. It's just a higher level. So it maybe takes a little bit longer, okay, to upregulate. This ratio is still coming down, okay, less dead space, greater depth of breathing, response is coming down, tidal volume is going up and then plateaus. Frequency is not necessarily linear, but overall we're seeing an increase in the frequency of breathing. So resting, we breathe about 12 times per minute. Moderate aerobic exercise, we're breathing, looks like maybe around 35 times per minute. So, but we're still within the aerobic zone. Okay, external respiration in the lungs, same pattern, right? So alveolar ventilation, go up. What we get here is just a little bit of a rise here after about an hour. That's caused by an elevation in body temperature. So especially if we're working out in a hot environment where we're losing a lot of body water, okay, where we become a little bit dehydrated, and that tends to increase our breathing. Alveoli versus arterioles, okay, so there's a difference there, okay, just like we talked about before, but it's for the same reason.
So in the first one, sorry.
Go ahead.
So for the longer, like the increase over time,
You mean this part right there?
the y-axis, the ventilation of alveoli, that's just increasing, like just the rate of it, or the
It would have to do with your depth of breathing would be increasing. So remember, alveolar ventilation is like ventilation, only we're subtracting out the dead space. So overall ventilation is just tidal volume times frequency. Alveolar ventilation is just taking into account the air that participates in gas exchange. Okay, so the dead space, how do we define that? Air that does not. So we're just subtracting out the dead space from the tidal volume and then multiplying that by the frequency. So that's the only difference is alveolar ventilation, we're just taking out the dead space and just considering the liters that are participating in gas exchange. Does that help? Mm-hmm. Okay.
Thank you.
Okay, internal respiration at the muscles. So the only difference is we have a much larger difference in oxygen content between the arterial and venous side. So all that means is we're just taking up more oxygen at the tissues. So you bring in a certain amount of oxygen via the arteries, arterioles. You take up the oxygen in the tissues, and then you have what's left over in the veins. So all we're saying here is there's just a greater difference. The venous saturation of O2, same thing. You're going to expect a drop because you're taking up more within the tissues. Okay, so very last category here. Okay, so aerobic testing to maximum. Okay. So we have a few things that are different. Okay, so I want you to look at pulmonary ventilation to start off with. So we're going from a resting state all the way up to a maximal state. So we see a couple of what we call break points. So let's take a look, this graph to start with. So we're going from a 0 workload to 100%. So this would be your maximum over on this side. So up here with your ventilation. Okay. Let's see if I can get this to come up here. There we go. Okay. So the first break point, okay, that's VT1, has been called the point of optimal ventilatory efficiency. So efficiency. So efficiency is basic in a general sense is doing more for less. Doing more for less. So in this case, it reflects the point at which maximum amount of oxygen can be taken up with a minimum of ventilation. Maximum amount of oxygen can be taken up. So oxygen consumed for a minimum of ventilation. So that's efficiency, right? Taking in the most oxygen we can for the minimum amount of ventilation. That's right here. So at roughly, what is that? That would be maybe a little over 60% workload is where we get that optimal ventilatory efficiency. Okay. All right, so the second break point is often called the respiratory compensation point. So we're going right here, and then ventilation really takes off from that point. So you're going along, it's pretty linear until you reach that first point, and then it starts to increase a little more, and then you get here and it just skyrockets. The liters of airflow would disproportionately increase. So this is what we call a relative hyperventilation. So the reasons for that, now there's, if you look at your book, there's debate on this, but this point right here is often associated with the anaerobic threshold. Okay, anaerobic threshold means we're getting supplementary assistance from the glycolytic system. What's the end point of the glycolytic system? If it's lactic acid, what happens to lactic acid? Like we talked about a few minutes ago, right? Lactate and hydrogen ions. What do hydrogen ions do to respiration? Do they affect respiration? Yes, because what hydrogen ions means what for pH? pH drops, which does what to respiration? Remember? What are the 4 key factors? Reasons, okay, decreased pH. Okay. So, tying this back together, right here. Yeah. So that could be associated with the anaerobic threshold because there's a connection between what your muscles are doing and respiration. So if pH is dropping a little bit due to hydrogen ions, that's going to stimulate more frequent breathing. So this is maybe a rough indicator. And again, I love this author because there's scientists on different sides, and on so many issues, this author really tries to balance and give each group their say. But overall, this could represent the point where you're starting to cross over into that anaerobic zone. So you get to this point, and how much longer do you think a test is going to last? Yeah, about 2 minutes. So you know, and you can see it because the software on the K5 system will show both of these breakpoints. You know that once you cross that, you're probably looking at someone that's going to be done in about 2 minutes because they're in that anaerobic zone now. So the other ones, do you notice any change really in the shape of? It's still the same. The ratio is going down. Depth of breathing is going up until right at the end there, and I'm not sure what's causing that, whether there's some inhibition or maybe it's just the high frequency is kind of taking over to control the increase in the ventilation, probably so. But overall looks very much the same, just higher level. So how can we use this? So you're an elite endurance athlete and you're coming into a lab to talk to an exercise physiologist about all of these respiratory and cardiovascular variables. So you're trying to get the edge in your training. So the pulmonary ventilation can serve an important purpose. So let's see what the practical application is. The workloads at which the VTs occur are related to endurance exercise training and performance. So VT1, okay, so that first break point is thought to indicate the upper boundary of moderate exercise. Upper boundary of moderate exercise. So that'd be so for an endurance athlete, that'd be the long, slow type of training runs. Long, aerobic type exercise. VT2 is thought to be heavy, but not so heavy that you can't sustain it. So VT2, that workload that you're getting at VT2, which is right around, looks like about 80%. So you'd probably be correlating this with a certain heart rate, because outside when you're training, you can measure workload in a lab, but you can't really do that when you're outside in a practical setting. So you'd probably be looking at, okay, at this workload, your heart rate is right here. So outside, you'd say, okay, when I reach this heart rate, I know that I'm at this workload, which means I'm just about at that second break point right there. So you'd kind of be correlating that with heart rate because it's easy to measure. And so you go past VT2 and that's where it's going to be not sustainable. So this means that VT1 and VT2 can be used for exercise prescription. Training at work rates between the two would stimulate the best gains in aerobic fitness, right? So you're getting the most benefit from your training time if you know the workload and heart rate associated with these 2 points. You know you're in that zone where you're going to be seeing gains in your VO2 max. Okay, external respiration. Okay, happens in the lungs, right? Gas exchange. You notice any difference here, really? Alveolar ventilation, you'd expect it to increase. Okay, you've still got those 2 break points because it's maximal. PaO2, alveolar PO2 versus arterial PO2. Okay, this is where we have another difference. So you notice as we get past about 60%, maybe even less than that, about 50%, we get this response. It's called EIAH, and it begins to drop. The saturation of oxygen as well as the arterial pressure of oxygen begins to drop once we get to a workload just below 60%. So what do you need to know? You need to know what EIAH stands for. Okay, so it stands for exercise-induced arterial hypoxemia. Exercise-induced arterial hypoxemia. So I noted the page number in your book there. The causes of exercise-induced arterial hypoxemia. So overall, it's caused by a faster transit time of red blood cells through the pulmonary capillaries. So blood leaves the right side of the heart, right ventricle, through the pulmonary arteries. Pulmonary arteries eventually become pulmonary capillaries that surround the alveoli. And so this is where we have a faster transit time of red blood cells through the pulmonary capillaries. Why is that a problem? Blood cells cruising through those pulmonary capillaries faster than under resting conditions.
Less time for the oxygen to be picked up?
Yes. It's a mismatch between perfusion and ventilation, right? Remember way back, first part of chapter 9, we talked about the ventilation-perfusion relationship. Ventilation is airflow, perfusion is blood flow. So if we get to certain workloads where blood cells are going too fast due to greater pressure from the heart, that's going to reduce the amount of time for oxygen pickup. So there's less time for equilibrium in gas pressure between the blood and alveoli. So exercise-induced arterial, what's the last word? Hypoxemia. That means low oxygen. Hypoxemia means low oxygen. So in normal sedentary or moderately trained people, okay, like most of us, pulmonary capillary blood volume increases with exercise. What does that mean? That means there's more blood going where? To the pulmonary capillaries, right? So that just means we're using more of our alveoli. There's more blood going into these pulmonary capillaries that surround the alveoli because we need more oxygen between rest and exercise. That's all that means. So the red blood cells are going through in a time that's sufficient to allow for gas exchange, equilibrium of gas pressure. Okay, so now we come to these highly trained athletes, these rare people, okay? Things are different for highly trained athletes. Why? Because they push their bodies to the absolute limit. What a human is capable of doing, they push those systems to the limit. And so this is where we can have a mismatch. Okay, so when these athletes continue to increase their workloads and cardiac output, okay, so blood coming from the right ventricle to the lungs, pulmonary capillary volume cannot expand further. So they're trying, their right side of their heart is trying to get more blood to the lungs, but they have a fixed amount of capillaries. And so what's the solution to this problem with a fixed amount of capillaries that you're trying to get blood to go through? What's the solution? Say it louder. Go faster. So if you have a fixed amount of capillary volume, but you're trying to get more blood flow through that fixed amount of capillaries, the solution is to speed it up, right? So the pressure from the right side of the heart is going to go up to push that blood through faster to compensate. So blood flow velocity increases. So there's not as much time to pick up oxygen before that blood gets back to the left side of the heart and then pumped out to the body where the muscles need it. So considerably less than that required for gas equilibration. Does that happen with all highly trained athletes? No. This is sometimes. It's not something that you can always count on happening. Sometimes. Okay, internal respiration, maximal exercise, AVO2 difference. Okay, you're going to increase until you can't consume any more oxygen at the tissues. So we go up, and then right there. So you could say you're getting to your maximal VO2 at this point because your body's not consuming any more oxygen. You're starting to plateau out in what your tissues can consume, so that you have the same amount left over in the veins because your tissues can't take any more. Saturation, can you just go down and then plateaus just like you see here? Okay, static exercise. What's static exercise? Yeah, tension. Are you still breathing? Yeah, so Bryn, when you do all those planks, are you still breathing? Yeah, of course, right? Try to. Okay, that's a skill. You learn. You learn to tighten, and you're still breathing. Okay. However, the muscles that are tightening are compressing capillaries, which means you're not getting blood flow out of the muscle. What are we producing in muscle during the course of those planks? CO2, which does what? Stimulates. It's the most important factor, right? So you're done with your plank, that muscle relaxes, and all that blood flow that's filled with CO2 is going to do what? This is going to cause a spike in your ventilation. Just this transient, where it's going to last for about a minute, you get this, we could say, a relative hyperventilation caused by the blood that was trapped in that muscle that was sustaining tension for a period of time.
You said it's trapped, so the CO2 can't leave it?
It's because the blood would be trapped because the capillaries are compressed.
Does that mean it can't get O2 either?
Yes.
Okay.
Yes. So just the muscle that's under tension.
Okay.
So that muscle would be emphasizing anaerobic metabolism for that period of time. But then you release it, and what happens? So then all the blood flow is then restored, but you've also got to get rid of that blood that had high CO2 and hydrogen ions and so on, which will then make the circuit and cause an increase in this ventilation for a period of time. You can see rapidly after, though, it goes back to baseline. Okay, so I asked your coach about breathing and what he does, because he's had some very, very successful, awesome distance runners. So you can read here, breathing isn't something that I usually coach unless we notice someone seems to be breathing excessively or at an easy pace. In these cases, I suggest trying to have a rhythm with breathing. Okay. Three steps running, breath in, 2 steps running, breath out. Usually in through the nose, out through the mouth. I'm going to try that. In through the nose, out through the mouth. Then as the pace quickens, then trying to have a 2-to-1 ratio. Only time going 1-to-1 is at the end of races when effort is at its highest. So some excellent practical type of advice there. So there's a lot to this. I'm sure that I'm wondering if this helps reduce side aches too, because maybe it's just because I'm out of shape, but I get those when I run. So I'm wondering if trying this would help with that. Okay, so I want to take just a few minutes. Let's watch that little video clip on increasing oxygen consumption.